Nick Petrick from the FDA.
Hi, so I'm going to talk about assessment of quantitative. I'm going to give you a sort of a brief overview of, and I have no disclosures. I'm going to give you a brief overview of medical device regulatory framework, and I'll try to do that in a general case. I'll talk a little bit about software and where we're going. I'll just give a real brief introduction to that, not all the details of what we're trying to do. And then I'm going to talk about just one sort of quick example, talk about quantitative imaging tool assessment. I'm going to talk about a single feature analysis and just show you sort of what we do. It would be applicable in the general sense to a multi-parameter analysis as well. But I'm going to just show you some of the issues that come up even for a single feature that seems like it should be simplistic. So just a quick overview. I'm from the Center for Devices and Radiological Health. Our role is to protect and promote the health of the public by ensuring safety and effectiveness of medical devices and the safety of radiation-emitting products. We have sort of strived to speed the transition of innovative and safe and effective products to market throughout the product life cycle. So just trying to show you a graphic of the product life cycle. We may not be involved in the discovery part, that may be up to the companies, but we, especially in the device side, try to be involved in the preclinical development, preclinical testing and clinical testing that's associated with the device. We're obviously involved in the regulatory submission and decision-making process. Once a product's on the market, there is potential for this post-market compliance and surveillance that occurs. And what we're trying to do, especially when we're talking about AI and different types of algorithms, is this sort of feedback loop when you're in the clinical space, that there's data that comes back and potentially helps you to do new discovery, and we go back and iterate through that process. So just some general idea about device classes and pre-market requirements. There are 3 different classes of device: Class 1, which are the lower risk; Class 2, medium; Class 3 are higher risk devices. There's controls. I'll tell you what these are specifically in the next slide, but just the general controls for Class 1, general and special controls for Class 2, and general controls and pre-market approval forms for Class 3. And then the process is typically for Class 1, they're exempt, so we typically wouldn't see data associated with that directly. For Class 2, there's something called a pre-market notification. That would be a 510K, or there's something called a de novo pathway, which I'll talk about a little bit too. And then for Class 3, it's a pre-market approval. So general controls, these are controls applied to all medical devices unless exempted specifically by regulation. They are things like registration and device listing, adverse event reporting, good manufacturing practices, and there are a number of others that go along with that pathway as well. Special controls are really the controls that are needed beyond these general controls, and they're usually device specific, and they include things like post-market surveillance, special labeling that goes with a particular device. It also includes pre-market data requirements. So this could be you need to do a clinical study and a pre-market requirement for different types of devices. So it depends on what the scenarios are, what might be needed for different types of devices. How things come to market in the US. The 510K pathway is used in Class 2. It's the most common pathway for medical devices. That's to demonstrate substantial equivalence to something already on the market. So there's a device on the market, it's in Class 2. You're trying to show you're, in the FDA terms, substantially equivalent. And that's not, when the law was actually developed, it was those terms were used because it was a term that wasn't defined anyplace else, and it allowed this sort of flexibility in what that actually means and how it should be implemented in the regulatory framework for devices. The other pathway is what we call de novo. This is for devices that will go into Class 2, but there are no predicates available. So it's a new device, but there's no predicate available. There's enough special controls or general controls that can support it being in Class 2, so it becomes if it gets on the market, it becomes a predicate for all the other devices and stays in Class 3. And a PMA is what you have to demonstrate a reasonable assurance of safety and effectiveness for that device. The other area I just want to make sure people are aware of, we have something called Q-subs or pre-submissions, and we use this a lot in the center, where companies come in with specific questions about their device before they're in the official regulatory pathway, and sometimes it happens while they're in that pathway, but usually before they're in that pathway, where they have an opportunity to ask specific questions and get specific advice. Typically, this device is not going to be binding on us necessarily, but of course, we're trying to give useful information for them to conduct the right studies or to develop their indications for use that make sense or appropriate for that device. So it's an important tool that everyone should be aware of, and it's utilized a fair amount of times. So just some ideas of what types of devices fall into different categories. I just throw you some examples: stethoscopes, tone depressors, manual wheelchairs. Those are in class 1 devices, typically exempt. We're not going to see a lot of data associated with them. There are other ones that fall into that category as well. Class 2 devices, I come from a background in imaging, so most of your sort of standard imaging processes, CT, MR, ultrasound, those are all class 2 devices. A lot of those have been on the market for actually a pretty long time now. Most of the imaging computer-aided diagnosis and detection devices that are seen that are on the market, those fall into class 2. There are a few that fall into class 3, but mostly fall into class 2. A number of in vitro diagnostic tests fall into class 2 as well. So depending on what those tests are, they can be in class 2 as well. Class 3, things like novel imaging systems. When digital breast tomosynthesis came onto the market, it's a class 3 device. There are different requirements for that data associated with it. Things that are higher risk, leadless pacemakers, those are in class 3. Some therapeutic devices, those are going to end up in class 2 because there's a high risk associated with them. And then, of course, there are some IVD tests and computer software and computer algorithms that are going to fall in class 3 as well. So I'll shift focus a little bit and try to focus specifically on software. And there's a group, the International Medical Device Regulators Forum, which is a group that got together of international regulators to come up with a concept for how to deal with what we're calling here SAMD, or software as a medical device. And that group was intended, software as a medical device is software intended to be used for medical purposes without being part of a hardware medical device. So this isn't going to be a closed loop control system within a artificial pancreas. That's not going to be, this is going to be devices like AI applied to determine diagnosis of disease that apply to imaging data, or something that's independent of the device. This is going to include a lot of the artificial intelligence, machine learning applications that people might be thinking about. Not all of them, but it'll encompass a fairly large broad. But it's also broader than that. It can include data for organizing data and doing other things within the clinical framework as well. The outputs of that, they had a number of outputs from this group. What I'm going to concentrate on is that final one, that clinical evaluation. This is particularly important because FDA adopted this particular document as guidance in 2007. So FDA intends to consider principles of the IMDRF report and evaluate in involving our approaches to AI and machine learning. And that's more specifically, but in general, software as a medical device. So what are the basic principles? And these probably are not anything that's particularly new to everyone in this room, but here are the concepts. First of all, clinical association. Is there a valid clinical association with the software as a medical device to a clinical condition? And sometimes this is trivial because there's all kinds of literature, there are professional guidelines already available. Other cases, it's sort of stretching the clinical framework, and so there may be a need for secondary data analysis or clinical trials potentially in order to just show that clinical establishment. The next part of it is analytical validation, again, somewhat parallel for IVD types of approaches. Does the software meet the technical requirements? That would be both providing evidence of the software correctly is constructed, so that's sort of the validation side of the software side. The other part is demonstrating it meets the specifications according to user needs, which may require some type of actual validation data to support that as well. And then the final part is obviously clinical validation. The evidence that shows that the SAMD has been tested in the target population and has clinical meaningful outcomes associated with it. So those are the basic principles of the IMDRF policy. That's the basic principles that FDA has put into guidance as well. So some of the new approaches. So we've been dealing with software as a medical device, especially in imaging, for a long time. 1998 was the first time we had a breast CAD come onto the market in my group. There might have been some other software before that. I wasn't an FDA then, but so it's been around for a while. So we have a pretty good approach to doing that. Of course, with the adaptation of deep learning and other possible approaches, we see that's in commercial product. There's this idea that there's going to be a large increase in the number of these tools that come along, and that's what we expect. We're looking within that IMDR framework of sort of readjusting how we might approach software as a medical device and looking at the risk associated with it and trying to make determinations of what types of software really do we need to regulate or see data on. So we may regulate it in a general sense, but we may not need to see data on just different types of software. So systems that display images, do we need to see data that they can display images correctly? Maybe that's not necessary in all scenarios. So as the risk of these devices get higher, as we're talking about decisions associated with diagnosis or treatment of devices, those are the types of data that we'd expect to see independent review on. Somewhere in the middle, there's this threshold between what types of data we might see data on, especially performance data on versus the types that we may not need that for different types of software as a medical device. Again, there's a huge spectrum of possible information that we can get out of these particular tools. And those can extend from clinical practice, things done for the clinician, to software that's used at home for certain individuals, going all the way to these sort of health lifestyle types of data about heartbeat and heart rates and other things that are out there that sort of bridge the gap between whether they're really medical devices or other types of consumer information. So just some takeaways from the device regulatory path. There are 3 tiers of devices that we evaluate. The indications for use and the type of technology are equally important for deciding what validation data is needed. So it's not just that here's my device and this is what it does, but what are the actual intended uses? What is the population that's going to be applied for? Those are really important questions and obviously have a large impact on the type of data we might see. And then there's this process of Q-subs or pre-submissions where companies have this opportunity to ask questions and get feedback before they delve into a large clinical study potentially and potentially get in the wrong pathway. And then just around AI tools, machine learning, we have substantial guidance already on this area, especially related to imaging, but in general as well. But with that being said, we're trying to evolve our approaches and come up with new concepts of how to deal with especially what's expected to be a large influx of these types of tools. And I told you about sort of the IMDRF perspective. There's pre-certification. There's a couple other programs that are going on as well in parallel that may adjust the way we approach these. So the next part I'm going to just concentrate and go through quickly is this quantitative imaging tool assessment. What is quantitative imaging? This definition is from the Quantitative Imaging Biomarker Alliance. It's extraction of quantifiable features from medical images for the assessment of normal or the severity, degree of change, or status of disease, injury, or chronic condition relative to normal. So again, you can talk about this as a single feature, and I'll talk about volumetrics as one feature, but it could be multiple features, AI, combinations of feature from imaging and non-imaging. The general process is going to be somewhat consistent. And we're currently working on a particular guidance associated with sort of these single biomarker types of quantitative imaging measurements. And I just tell you right now, if a company comes in and says, I have this tool that can measure the size of a lesion, as long as that's under the clinician's control and there's no claims associated with how well that performs, we don't see data on that. It just goes onto the market. It's a tool, it's in the packs, and it's used, potentially used every day, potentially not used at all. And you can run that across a gamut of 100 different tools that are currently available clinically. What we're looking for is we want to get to the point where these can have claims associated with them. So what can they do? Why is this measurement useful? And then potentially, what are the error bars? What's the performance of that assessment? Not just a number put out there that clinicians are supposed to accept or not, but here's a number and here's a context for that number. So here's just a really simplistic approach. This isn't a particular tool. This is something we've worked on a long time for research inside FDA and with the Quantitative Imaging Biomarker Alliance. But just CT lesion volume is a quantitative measure of actual tumor size in vivo. Simple, relatively straightforward. There would be particular claims of how well we could do that. What is the confidence around that? I'm not going to go into all those details. And what we're going to look at in this context is not just the performance of a single marker, although I'll show you results from part of that analysis on a single marker, but what happens when you have 10 of these, because there are 10 different companies produce them, and they're used in different clinical sites, and we know from trials, it's hard to get the same imaging system on the same study. It's hard to get the same tool utilized in 2 different longitudinal settings potentially. So what's the consequences of having that? So the basic components, again, pretty straightforward. We have some verification validation, software testing. So does a tool meet the technical specifications? That's the software side of things. And then we'll talk about analytical validation, or what I'm going to call technical assessment, which is how well can we make the measurement? How do we go about measuring how well we can make the measurement? There's a whole other side of this, which I'm not going to talk about here, is the clinical assessment. This typically requires some sort of randomized clinical studies to associate. So I can make this measurement of volume. I can make this measurement of size. RECIST is always commonly used. There's really no studies that show RECIST has this particular clinical context. It was a measure that's logical. It was sort of made in a framework that is logically makes sense, but is 30% the right number on the RECIST criteria? That hasn't been clinically validated, although it's used, and we're going to live with that measure. This requires more data and a more complicated design associated with it. So I'm going to talk about just 2 different parts of this, accuracy and precision. Same basic statistical concepts that have been talked about before. For this particular case, when we're talking about accuracy, one of the problems in imaging studies is we don't have a reference for in vivo clinical studies. What's the size of a lesion in a CT image? I don't know. I can get 5 radiologists. They'll take a look at that. And depending on how complicated that region is, we could have variability of 10%, we could have variability of 50% in that measurement. So what's the real truth? Well, we don't know that. So if we want to understand bias and linearity or accuracy, we want to talk about, is there ways of doing that? And here's a surrogate for doing that. I'm not going to say this is the perfect approach to doing it, but we can use phantom studies. In this particular context, we'll look at a thorax phantom with synthetic nodules. So linearity is going to be a measure of how change in the reference reflects proportional change in the measurement on average. So we're going to plot the measured value versus the true value. So when we're talking about linearity, don't confuse that with the linearity of the algorithm or the performance. This is statistical linearity associated. How well does my truth represent, how well does my measurements represent the true value? So if we have proportional change, we have a linear association. We can talk about this case where we have a slope of 1, that's a linear association. We can talk about a different slope, that adds some more complication, but it's still a linear relationship. And of course, we can have some sort of nonlinear relationship. And what we want to see is a linear relationship. And if we don't have that, we want to decide what range is linear enough that we can deal with that. We also want to talk about bias. This is going to be the difference between the estimator's expected value and the reference standard. And there are different types of bias. We could have unbiased measurements, so there's no bias. We can have a constant bias. That's useful in the sense it's a bias, it exists. If we can measure it, we're all good, but that's not always what we can do. But if we know we're making differences in measurements over longitudinal measurements like size, we might know that if the bias is constant, we at least know the difference in the bias between two different time frames, so the change is going to be unbiased estimate. And then, of course, we can have non-constant bias, which adds a little bit more complication for how we assess this. So here's just a simple phantom study. We had 4 different sizes ranging from 5 millimeters to 20 millimeters. We can conduct a more complicated analysis to decide whether a linear model fits or not, but in this case, here's a linear model. We get a slope of about 0.96, so almost a slope of 1. It's pretty consistent, has a pretty good goodness of fit to it, so that's our linearity measurement. You can see at the bottom we calculate the bias. We have a 39% bias. This is a highly biased estimate, but we have an estimate for what it is in these phantom experiments. The second part we'll talk about is precision. And in this case, we want to look at the clinical context. Looking at the phantoms in precision is even more problematic than looking at accuracy and linearity in a phantom experiment because of the variability in the patient. So what we've done here is done something called a coffee break experiment. The patient was scanned twice within a 15-minute interval using the same scanner and the same reconstruction. So this is sort of like a best case scenario because that's typically not what's going to happen. But let's assume that's what data we have. We're going to go look at clinical repeatability. So variability under near identical conditions. And in this case, we're going to assess individual algorithm performance. So we're going to have one single volumetric algorithm, look at how repeatable it is. In this case, we're going to use a repeatability coefficient. Just keep in mind that a smaller value of repeatability coefficient means more precision, higher precision. Second is we're going to look at clinical reproducibility. Again, that's variability under a range of experimental conditions. Our range is just going to be across multiple algorithms. So we're just going to look at that one variability and range. We could talk about different variabilities as well. That's going to be the reproducibility coefficient. Again, smaller value, the better. So here's just a single algorithm repeatability. You can see that it's pretty broad, right? We have 100% for algorithm 3. That's not very good. But we have a range of these sort of best performers that are on the issue, a range of 13 to 14%. So you might say, how well can we interchange algorithms and utilize this across different algorithms? And I'll look at 2 different things. All the algorithms except for that 3, which is really large, versus all the better performing algorithms. So in this case, if we look at all but 3, the reproducibility is 84%, so pretty broad, right? So there's a big error associated with these measurements when we're using multiple algorithms, this wide range of algorithms. If we use the best performers, it's still 58%. So this tells us something about how we need to control our algorithms, our imaging in a trial in order to achieve whatever performance we need. If we need to get down to 13 or 14% of performance, we need to be using the same algorithm at every iteration of the analysis. If we could live with the 50 or 60% variability associated with the algorithm, maybe all we have to do is control the imaging and we can be okay. So this is where this idea of what the effect size is relative to what our variabilities are important. What we need to do is be able to measure them, understand what we can control, because in clinical practice, it's going to be hard to control the scanner everywhere. It's just not very realistic. So what can we control? And then what are the variabilities we can live with? And then what effect sizes are enough that we can see differences? So just a quick summary. Again, devices are into 3 tiers. The indication for use and the type of technology are just as important as in deciding what the validation method is needed. And again, we have these evolving approaches to software as a medical device. And then as radiomics or QI tools or any type of different combination of information, we're looking at verification validation, which includes technical assessment or analytical assessment, and then clinical assessment of performance. So, and I'd like to thank my team who did a lot of the work on that, especially that quantitative imaging example. Thank you.
Thank you very much.